Okay, everyone, I think we are ready to start. Good afternoon. It is really my pleasure today to welcome Dr. Bansma to give a guest lecture in our course. Dr. Bansma is a staff physician and a scientist at the Hospital for Sick Children here in Toronto. He obtained his PhD in nutrition and metabolism, as well as his MD degree in the Netherlands, and which was followed by training in various cities in Europe. And then he held a position as an honorary lecturer at the University of Malawi, and also he was a staff physician at the Queen Elizabeth Central Hospital in Blantyre, Malawi, which I guess is where he is today. At the Hospital for Sick Children, his research program focuses on understanding the regulation of nutrient metabolism. The ultimate aim in his lab is to develop novel but simple interventions that are evidence-based and that ultimately can help reducing mortality in malnourished children. And Dr. Bansma, thank you so much for giving this lecture while being so far away. And thank you for your time. And we are really looking forward to your talk. You should be able to start sharing. Let me know if any issues. And I think Dr. Bansma will be happy to answer questions. If maybe we can save them for the end. Dr. Bansma, whatever is easier.
I don't mind, actually. I'm perfectly happy for people to ask questions throughout, if that's what they want. And,
okay, so that's great. So let's keep it let's keep it like easygoing, everyone. And if you want to ask a question, you can use the reactions and also just go ahead and ask the question as you would do in the class. And we are not recording this lecture, okay, because there is a question that has just been asked. So thank you. Please, you can go ahead.
Okay. Let me just so many things open. One second. Yeah. Okay, so you all see my slides, I hope. If not, please let me know.
Yeah, all good.
So okay, great. So sorry I'm not here in person. Normally I give this lecture at the Department of Nutritional Sciences, but unfortunately I'm in Malawi at the moment. So that's why I'm doing it remotely. I could have asked you all to come down to Malawi with me if Dr. Kameli would have paid for it. I would have gladly take you all along. But unfortunately, that didn't happen. So that's why I'm remote.
Dr. Kameli is poor.
So what I'll do today is to really talk through the relationships between nutrition, microbiome, and growth. So how nutrition can affect the microbiome and how the microbiome can subsequently affect growth in children. So I have some learning objectives. So I have 3 learning objectives. The first is to demonstrate relationship between the microbiome and growth. And secondly, to explain the relationship between micronutrients and intestinal microbiota. And finally, I will also talk a little bit about, because that's still early stages, about potential treatments to improve the microbial dysbiosis that we see in children with malnutrition, which is the population I really focus on for my research career. Okay, so let me first start by giving an introduction to how common malnutrition is around the world, and some of the history of and the trajectory of the prevalence of malnutrition over the last couple of decades. Because I think it's important to first give you that perspective, which will then explain, I think, why people around the world have invested their time and resources to try to address this problem. So this is some data. This is jointly from WHO and UNICEF and the World Bank. Every year they report their global estimates of stunting, wasting, and also more recently on overweight in children around the world. So on the left, you see the actual percentage, and on the right, you see absolute numbers in millions of children. So the yellow-orange line represents children with stunting. So you can see that the percentage of children with stunting has decreased from around 30% to 20% over the last 20 years. So that's a significant drop. And you also see that in the absolute number. You have to, I think, put it into perspective of a growing population, right? We've grown significantly over the last 20 years. So even with this population growth, the burden of stunting has significantly been reduced. In part it's related to the development, especially in China, which has lifted, I think close to 800 million out of poverty in a relatively short time period. And that, of course, has had a significant impact on stunting. But also other countries have made significant progress in reducing the burden of stunting. But it still means that, 20% of children around the world suffer from stunting. So they're short for their age. They have low height for age. The data on wasting, so being thin with, i.e. a low weight for height or a low weight for length for young children who can't stand, that burden is around 7%, and leading to about 45 million children around the world. What you can also see is that the burden of being overweight has slowly increased over the years, less so in terms of percentage-wise, but because of population growth in absolute numbers, it has. Okay, so now you know how common these nutritional conditions are. So this is some data on how wasting is, where wasting is most prevalent around the world and where it's least prevalent. Of course, as you can imagine, in the high income settings of Western Europe and North America, the prevalence of wasting is very low. There's actually no data, but the prevalence is supposed to be reportedly very, very low. But also in Central America, South America, the prevalence of wasting is quite low. Similarly with China. But what you can also see on the other hand is that the burden of wasting really lies in Sub-Saharan Africa and Southeast Asia, mainly India, which you can see is colored in the red, or in some certain areas, more than 50% of the children are wasted. And why does that matter? Why do we care if somebody is thin? Why do we care if somebody is wasted, has a low lean body mass, and or low fat mass? And that's related. This is an example. So this is some work that I've been part of as part of a very large international network, whereby around 3,000 children were followed up during their hospitalization with any acute illness, and then were followed up for about 6 months, or 180 days. SAM stands for severely malnourished children, severe acute malnutrition, although the acute has been, WHO I think is revisiting the label of acute because many forms of malnutrition are not acute. MAM is moderately wasted, and the non-malnourished are non-wasted children. So what you can see is that, the initial slope, because in the y-axis, that's you see mortality. So 0.2, for example, it means 20% of the children have died. So you can see in the severely malnourished population, after hospitalization for any acute illness, it can be pneumonia, diarrhea. There wasn't really a clear relationship with the type of illness they were admitted for. 20% of those children will have died by 180 days. And you can see that the slope of the curve is very steep in the beginning, and then it levels off, but it never goes to zero, or it levels up where it's completely flat, right? So you can see that that vulnerability remains. And actually what we found was, because hospitalization was around 5 to 7 days, that about half of the mortality actually occurs after discharge from hospital. And that was also true for moderately malnourished children, and the same for non-malnourished children. And the increased risk between non-malnourished and severely malnourished was close to was about 8-fold, 7 to 8-fold higher. So why is that? Why are these children so frail? Why are these children so vulnerable to, simple acute infectious illnesses that other children recover from quickly? And that's related to the fact that malnutrition affects many different organ systems in the body. I think almost every organ system is affected. So neurologically, there are symptoms, there's lethargy, irritability, there's some data on changes in serotonin levels. Cardiac function is potentially impaired, although the jury is still out on whether that is true. There's hepatic dysfunction. So the hepatic enzymes that are produced, or the hepatic bile that is produced to help digest lipids and help with lipid absorption, is impaired. And similarly, the exocrine pancreatic function is also impaired. So digestive enzymes that are secreted by the pancreas are reduced. Hormonal systems are affected, the immune system is affected, and the gut is infected. And we'll talk a little bit more about the gut later on. Again, if you have any questions, just please let me know, right? I see some things in the chats, but as I'm presenting, it's difficult for me to follow the chat. So just speak up and then I'll hear you, and I can interrupt the presentation and address your comment or question. So this is how stunting is around the world, and the places where stunting is most prevalent. And you can see almost a very identical pattern as we saw with wasting. So Sub-Saharan Africa and India, Bangladesh, Pakistan, those are real high prevalence countries. But also Indonesia, as you can see, is quite clearly affected more than other countries. And this is overweight. And what is interesting with overweight, it's commonly being assumed to be highly prevalent in high income settings. But what we're seeing increasingly is that in countries that have made developments in their economic productivity, and their GDP has increased significantly over the last decades, that actually we're starting to see more and more obesity and overweight and obesity happening in those populations, including in children. And actually, in absolute terms, it's projected that soon countries like India, China, and other low-end or middle-income countries will be harboring the largest population of overweight and obese children. So no longer the high-resource countries. Okay, moving on. So now you have a little bit of background. So now we can move on to examining the relationship between diet and the microbiome. So there's some early work that was done about 10 years ago. That was done by a group where a really elegant set of studies whereby they fed people over the course of 10 days different diets. So they switched from a plant-based diet to an animal-based diet. And these are fractions of specific bacteria and fungi. And you can see how many of these bacteria and even fungi changed over the course of the switching between a plant-based and an animal-based diet. So this study really illustrated that even when you're an adult, you should have a stable microbiome. After a short change in your diet, you can have, within a couple of days, changes in your microbiome. And this explains a little bit the difference in some of the characteristics of the different diets. So of course, plant-based diet led to, that's in green, led to a much higher fiber intake and a lower fat intake and a lower protein intake. And the reverse was seen in an animal-based diet, right? So you can see here, so the extent of the changes in composition of the different diets. And what was really interesting that these changes led also to other markers of microbiome composition, including beta diversity. So the variability between subjects was clearly altered especially after switching to an animal-based diet, but also quickly went back to the original sort of state that these microbiomes were in these subjects. So this really was one of the first studies demonstrating, and that's why it was published in Nature, that the microbiome can change very rapidly upon changes in your diet. And then only a couple of years later, the first, actually there's one paper that preceded this, some work around malnutrition and the microbiome started to emerge. And initially that came really out of one lab, and they received a lot of funding from the Bill and Melinda Gates Foundation to really look at this, to explore this. And this is really the beginning, sort of early stages of the explosion in microbiome research, because it's not that old, only about 10 years ago that really that explosion started. Although the concept of a microbiome is much, much older. So this is based on a review that was published in Science, showing that if you have early perturbations in your diet affecting your growth, that will change your microbiome, and that will then have subsequently all kinds of effects on your health status, including metabolic, immune, and neurological effects. So this is one of the first studies, actually, I think the first, that looked at the microbiome in children with severe malnutrition. So there are actually two types of severe malnutrition. One is characterized by wasting, and the other is, what you probably remember from certain famines, is the disease entity called kwashiorkor, or edematous malnutrition. So it's characterized by edema of the limbs, the lower limbs, and it can also extend to the upper limbs and to the face. Hair discoloration, loss of appetite, a big belly, and fluid accumulation, really. So what they did here is they followed twin pairs, and they followed twin pairs where who became discordant for the development of kwashiorkor. So one of the twins would not develop kwashiorkor, and the other would develop kwashiorkor. And then they collected stool samples and they looked at their fecal microbiome. And then in a really elegant set of studies, they actually used the stool from these children to do some further experiments in the lab. So what they did was they transplanted the stool, a little bit of the stool, in germ-free mice. And then they exposed the germ-free mice to either a normal diet or really a Malawian-based diet. So a diet that's deficient. So they use stool from malnourished children, and they use stool from the discordant twins who did not develop kwashiorkor. And this is what they found. So this is the data of the mice. So they put these mice on a Malawian diet, so a diet deficient in protein. And the blue are the mice that received the stool from the children with kwashiorkor, so really the edematous malnutrition, and the red from the control group. And what you can clearly see is that the weight drop was much more significant in the mice that had the stool from the malnourished children, from the kwashiorkor children. RETF stands for ready-to-use therapeutic food. That's used commonly in low-resource settings to manage children with severely malnourished, who are severely malnourished, to nutritionally rehabilitate them. I can give you some history on RETF perhaps at the end if people want to know about it. So it's a high caloric feed. So you can see that the weight catched up, but not to the same degree in the mice that had the poop from the malnourished children. And then again, they regressed back once put back on the Malawian diet much more so. Although at the end, they reached the same state here. Yeah, so this is real proof for a causal relationship, because these mice were treated the same. The only thing was the change in the microbiome, and that's what led to differences in their weight. So that tells you that the microbiome can have a direct effect on growth. They also looked at the maturity of the microbiome, which you can see here between the two, and this is over the different stages. So this is this was a marker of maturity of the microbiome, which basically is a measure of the diversity of the microbiome, because as a child and infant ages, the microbiome becomes more diverse. What you can also see is that with treatments, that maturity increases, but then also it seems to, although this was not significant, seems to regress. And I'll show you something else later. But of course, what's the black box here? Because how does the microbiome do that? And that's something where a lot of research is being done currently. And this is one of the examples that was done in the same paper. And of course, these microbes produce all kinds of, in their own metabolism, as they use nutrients and metabolize these nutrients, they produce metabolites, and they are sometimes secreted by the microbiome, or they are detected if you analyze stool from within these microbes. So what they found, there were also clear differences in the metabolites of the stool between the different groups. And this is something is potentially providing a mechanism, or one of the mechanisms, as to how the microbiome can alter metabolism of the host, so in this case of the child, and lead to changes in growth. But I'll provide a little bit more, I go a little bit more into this later on. So this is a follow-up study, or a study in a slightly different population in Bangladesh. This is in a slum area where they also followed up children and gave them RETF, that same diet to help them grow faster. And you can see here also that the microbiome had a relative immaturity compared to healthy children, which you can see here in the blue. And then it improved with treatments, but again, over time it regressed back. So it wasn't permanent, that change. But people have also done the reverse. So this is a study, was done around the same time by the same group, where they looked at people who were discordant for being obese. So instead of being discordant for being wasted, or in the previous study, kwashiorkor, they looked at people who were discordant for being obese. So one twin would be obese, the other would not. And what you can see is that if they looked at, took the poop from these obese or non-obese people, transplanted it into the mice on a regular diet, you could see that there was clearly more weight gain in the mice that received the stool from the obese subjects, and a higher change in fat mass. And you can see that here as well, compared to the lean twin stool transplanted mice. So again, clear evidence that there is a causal relationship, both that the microbiome can affect wasting, so impairment in growth, but also, increased weight gain. And then what they did is they put them actually on a rather healthy diet, so low in fats and high in fruits, and they housed them either together, the obese mice, the mice that had stool from obese subjects or from lean subjects, but even on that healthy, low fats, low saturated fat, high fiber, still there was a positive change in their body mass compared to the lean. So even in the most healthy circumstances, there was a clear effect of the microbiome. And what they then did is they also looked at when they co-housed the mice with mice from the other group. So basically then the whole microbiome got mixed up because there's coprophagy among mice. So they eat each other's poop. So if you put them in the same cage, their microbiome basically becomes one that influences one another. And that influenced then also that led to a loss of the changes in weight, in body mass. So how can the microbiome affect nutritional status? So theoretically, you can think about a couple of things how the microbiome can do that. I fear I've given you 4. There might be more that I don't know of, but these are 4 that's for where there is some evidence and that you can also theoretically potentially appreciate. So one is by affecting host metabolism, right? So I just showed you that if you analyze metabolites in the stool, there are clear differences. And these metabolites can also potentially affect metabolism of the host. They can also lead to potential changes in appetite regulation. Some of the appetite regulators are actually in the digestive tract, and some are, of course, in the brain, and they can be affected by the microbiome. Then the other way how it can affect nutritional status is by affecting energy expenditure, so how much energy one burns in any given state. And finally, change in the microbiome can affect gut health, and I'm going to talk about a little bit more, and immune function. And all those factors can potentially affect nutritional status. Okay, but everything starts with being born, or even before being born. So there's growing data that pregnancy or actually women, pregnant women, and the microbiome in pregnant women can affect the microbiome of the offspring. And there are various ways how that can be done through exposure to the microbiome in the stool and potentially also through the microbiome in breast milk. And if you have a healthy microbiome, it might lead to a healthy offspring. And again, if the mother doesn't have a healthy microbiome, that can then also affect the offspring in a negative way and potentially lead to, in this case, obesity as depicted here in this paper. But also postnatally, being malnourished can affect children, as I just tried to explain. And this is how the concept of how people are looking at it in my community, my research community, at that cycle of being undernourished and the role of the microbiome. So altered microbiome is thought to affect gut health. And what's depicted here is gut permeability. So a leaky gut, it can lead to malabsorption of nutrients, which can then lead to inefficient and insufficient dietary intake because nutrients are lost in the stool. There can be an effect then on immune responses, which can affect the susceptibility to intestinal infections, which can then further alter the microbiome. I have a somewhat similar slide, but slightly different later on. So this is how that cycle potentially can occur. So I put this up in an earlier slide, this environmental enteric dysfunction or environmental enteropathy. So that's a concept of gut health being affected by being malnourished or having a change in the microbiome. So environmental enteric dysfunction or environmental enteropathy is characterized by villus atrophy, intestinal inflammation, or increased intestinal permeability. I have a picture later on. And the pathogenesis is thought to be multifactorial. One is poor sanitation, leading in part to an increased burden of pathogens, malnutrition itself, and immune dysregulation, potentially related to the being malnourished. So they're all interrelated. So this is a picture of a healthy gut. So if you would look under the microscope, this is what you would see. So nice long villi that are important for nutrient absorption, right? Because it increases the surface area. So you have more surface area for absorption of nutrients. And on the right, you see a gut that's affected by environmental enteric dysfunction or environmental enteropathy. So you see that the villi are much shorter. So that's something you, for example, would also see in celiac disease, right? Gluten intolerance, you also see shorter villi. But in malnutrition and environmental enteropathy, you also see shorter villi. Another aspect is you see inflammation, which is characterized here by these infiltration with these T cells and macrophages. What you have is increased permeability. So you get a leaky gut. So these intestinal cells are kept together by tight junction proteins. So they form a seal, but that seal is disrupted in environmental enteropathy. And that can lead to translocation not only of the bacteria themselves, but also of some of the bacterial products, for example, endotoxins, and that can then lead to systemic inflammation or even overt infection, systemic infection, and even sepsis. So some of the mortality that children who are malnourished, that increased mortality, is thought to be related to that increased permeability of pathogens, of microbes across the intestinal barrier. So this is another way of presenting that cycle, that's a downward spiral, actually. That's more like it, but I presented it here as an infection cycle. So it potentially starts with susceptibility to enteric infection. So in many very low resource settings, for example, you have livestock, you have chickens, roaming around the house and sometimes in the house, and they poop. And if you have infants who crawl around on the grounds, around the house or the hut, they get exposed to the poop of the chicken, for example, or other livestock, and thereby can develop enteric infections. Of course, clean water is often challenging in many places around the world, so still, unfortunately, so that can also predispose to developing enteric infection. That in itself leads to intestinal inflammation and malabsorption, and that can lead to malnutrition and further immune dysregulation and increased susceptibility to further enteric infections. So that's how you can get this spiral, this circle. But coming back to, because one thing you could do is give antibiotics, right, potentially, in these children. But there is data to indicate that antibiotic exposure, especially in infants, can lead to changes in the microbiome that are longer lasting and can lead to changes in growth. So this is a concept for which there is some data, but of course this is very difficult to really prove causally. But there is definitely some and rather strong data that antibiotic exposure can affect growth. And we know that also, for example, from the agricultural sector, where antibiotics has been used, for example, in chicken and other animals to improve growth. So that's been a long-standing practice. So I think that community has known for a long time that by changing the microbiome through antibiotic exposure, you can affect growth. So in this paper, they argued that if you have a limited perturbation of your microbiome by perhaps not by limited antibiotic exposure, it might lead to overweight. But if you're heavily exposed to antibiotics, it might actually affect growth in the other direction. So this is then sort of here in a more schematic way that's argued. So if you have a strong perturbation, you get some really profound change in microbiome and loss of the ability to harvest all the calories from your nutrients through the microbiome, and you can have altered immunological signaling and function that can ultimately lead to underweight. Whereas if you have a mild dysbiosis through antibiotic exposure, it can actually lead to overweight. So again, this is to some degree a concept. Okay, so I think I hope I have convinced you that there's clear evidence that one, nutrition can affect the microbiome, and two, that changes in the microbiome can affect growth. However, I think it's also important to just briefly talk about micronutrients and in this relationship between microbiome and growth and nutrition, sorry. So there's very strong evidence that deficiencies of many micronutrients or specific micronutrients are associated with poor outcome in children and increased vulnerability, for example, to infections, including diarrheal infections and pneumonia. So, for example, vitamin A, zinc, and iron here are depicted here as three really strong or important micronutrients in maintaining optimal immune function in children, and deficiencies are associated with increased risk to infections and death from these infections. But what's important to know is that the microbiome plays an important role in micronutrient homeostasis. So one thing is that many microbes, specific microbes, play a role in the synthesis of specific micronutrients. For example, riboflavin, thiamine, and other B vitamins, and they of course these vitamins play a very important role in metabolism, and especially around the B vitamins around energy metabolism, and thereby you can see how they might also play a role in growth through their affecting metabolism. So there is definitely a strong relationship with many of these water-soluble vitamins and your microbiome and specific microbes affecting synthesis of the of certain vitamins. Also, for the fat-soluble vitamins, similar relationships have been found. So this is for vitamin K, A, and D. And what's also important that these vitamins also play a role. For example, vitamin A and vitamin D have been found to be very important in gut health, and also in that gut leakiness I talked to you about earlier. So you can see then how if you have a microbial disturbance, a strong microbial disturbance, it can lead to intestinal barrier dysfunction through by affecting their nutritional status and or inflammation, but also potentially by affecting their vitamin D status, so indirectly through affecting vitamin D status. This is a study that we performed, whereby this was a secondary analysis of a trial where children received micronutrient supplementation, and we looked at the effect on the microbiome. So these are the different micronutrients they received. So it's a multiple micronutrient powder plus or minus zinc. And what we found was that actually supplementation with micronutrients can actually also lead to dysbiosis and potentially, growth of microbes that can be harmful for the child. So there's, of course, people have thought that giving micronutrients to children that are at risk for micronutrient deficiency is beneficial, but there's also potentially a risk, especially if you're already malnourished and you're worried about, growth of certain microbes in these already not healthy guts. If you can then feed these microbes by giving these micronutrients, it can actually potentially do damage. It's been known for iron. So iron supplementation has been known by affecting the microbiome, because microbes use their iron for their own metabolism. So they basically feed on the iron and can thereby worsen clinical outcome by exacerbating intestinal infections. But there's now also some data that some other micronutrients, including zinc, can potentially do the same. But finally, I think it's important to start to think about, can we actually do something about this dysbiosis, these disturbances in the microbiome, to improve growth? So either restoring normal growth in a malnourished child, or reducing weight gain in children who are overweight or obese. So these are studies. This is one a double-blind randomized trial that was done in Niger, in about 2,500 children, whereby they gave them amoxicillin or placebo, and they looked at and this was in severely malnourished children, looked at their nutritional outcome 8 weeks later. There was absolutely no benefit to giving antibiotics. And then in a systematic review that was done in 2014 in low and income countries of other studies that had used antibiotics and looked at growth, the effect was actually quite minimal. Yes, there was potentially, an effect, but it was very, very modest. So only 0.04 centimeters per month in terms of height gain after antibiotics, and also weight gain was very, very limited. So that means that even though, there's a clear relationship with microbes and growth, affecting the microbiome through antibiotic use does not necessarily lead to better growth. So perhaps it's the wrong antibiotics, or perhaps we have to realize that it's more complex. And, what people have used in the agricultural sector with livestock is not so easily transferable to people that have a much perhaps more complex environment and many more factors that are influencing growth. And then a lot of people have devoted their energy and time in trying to see of improving the environment, the sanitation, the water hygiene, whether that improves nutritional outcome. And in these two large studies on child growth. So what if you instill, proper hand washing, improve the water quality, improve sanitation by installing toilets and proper sewage systems? Can you thereby improve growth? And that was, thought to work by, reducing the intestinal infectious burden / affecting the microbiome. I don't have any data slide for that because these studies have been really rather disappointing. So millions and millions of dollars have been invested into these studies, mainly funded through the Bill and Melinda Gates Foundation, but also supported by the World Health Organization, but they've been rather disappointing. So again, changing the microbiome can potentially improve growth, but it's not so easy. And just affecting water quality, sanitation, and hand washing alone is not sufficient to have a clear, strong impact on child growth. what about probiotics? And this is a really rather interesting study from about 5 years ago, whereby initially they looked at germ-free mice and looked at their growth. So you can see here that on the darker lines are germ-free mice. So these mice are sterile. They don't have any bacteria, so they don't have a microbiome. So this data really showed that you need a microbiome in order to have optimal growth. So if you don't have a microbiome, you can't grow as well as when you do have a microbiome. But what they did, they had germ-free mice versus conventionally raised mice, and they looked at their growth and hormone developments, and so including IGF-1, IGP-3. So those hormones are really important in that growth signaling, and that signaling to help the bones grow. So that hormonal axis is really important for optimal growth. So they saw in germ-free mice that yes, also there was decreased growth, but there was also that was related to decreased levels of these hormones, the hormonal axis. But then they did something interesting. They gave one specific probiotic, in this case, Lactobacillus plantarum, so Lactobacillus species. This is a rather complicated slide. So the top here is just a wild type healthy mouse fed healthy diet. And then below that you have a group of mice that received the same healthy diet, but with this Lactobacillus plantarum. In the blue dotted line, they also received Lactobacillus plantarum as a probiotic, but that Lactobacillus plantarum species was altered genetically, so it wasn't the same as the wild type Lactobacillus species. And then again, they had the germ-free mice with lower weight gain and also lower height gain. But then they gave these mice, exposed them to a diet that would impair their growth. And then the real remarkable finding came about is that if you give this Lactobacillus species, you actually have growth comparable to a wild type mouse that's not germ-free. But if you just give a disrupted, an altered Lactobacillus, you have much slower growth. And this again is under, suboptimal nutritional conditions, and the same with body length. So one probiotic species can affect growth. So you don't need, a complete alteration of your microbiome necessarily, but this was a real proof of concept that if you just have one change, you induce a change by just giving one specific probiotic, you can thereby affect growth in a positive way. And this shows the mice in photographs, and this is their bone length, so you can really see that their bone length is positively affected compared to the germ-free mice that didn't receive the Lactobacillus plantarum. Another group of metabolites or compounds which are called oligosaccharides, and they're actually quite prevalent in breast milk. They're the third largest component of breast milk, and they're small sugar molecules. And there are very many different kinds of these short sugar molecules, these oligosaccharides. Here you see a couple of examples of these oligosaccharides. And they've also been found to affect growth. So they can be seen as prebiotics, right? So they affect the microbiome because they are the nutrition of microbial species. So here they did something very similar to what I earlier showed with the transplanted the mice fed or given poop from from different children, either as wasted or healthy children. So here they use stunted children or healthy, normally growing children, and they gave these different animal models only a Malawian diet or a Malawian diet with these oligosaccharides, and then looked at their growth. So this is the blue is the mice that received these human milk oligosaccharides, where they found a clear impact a positive impact on growth in these mice that received this particular prebiotic through HMOs, and this is lean body mass. So a clear improvement in growth, and this is bone length. So different types of bones and the length of these bones. So real proper growth, including bone length. So of course, the formula companies picked up on that, and many formulas now starting to contain HMOs because it's now felt to be beneficial for infants. But it tells you how complex breast milk actually is, and how many things we still don't know about breast milk, because this is all very, very recent. And finally, there's more and more thinking about, can we actually change the microbiome by giving very specific diets? So instead of giving a probiotic or giving oligosaccharides or giving antibiotics or changing the microbiome with a specific medication, such as an antibiotic, can we actually with foods, rather than foods targeting growth directly through the caloric intake and the protein intake, can we actually affect growth through modulating the microbiome? So that same group that I showed you that did those earlier studies in Science and Nature in these children, what they did is they tested different foods in the animal model systems, and the ones that were felt to have the best microbial composition in these animal models, they were then tested in children. So they had different types of diets, for example, that contain more or less lentils and all kinds of variations in the foods that were tested. And then they treated children with that. And this is a study that was published just last year in the New England Journal of Medicine. So this is the MDCF2. This is one of these diets that they tested, and it was in a group that was wasted, right? A low weight for length. They were not severely wasted, but they were mostly moderately wasted. And RUSF is a standard supplemental feed. So if they gave this specific microbiome directed food, that weight gain was found to be larger than in the traditional sort of nutrition habilitation food or supplementary foods that was given. So this is where the field is now, and this is where the field is going to. And you can see that many of the really exciting research is actually coming from studies in lower resource settings, where the prevalence of poor nutritional status is really, really high. So this is really, really exciting for the field, but also for the community and for us clinicians managing children who are malnourished or stunted and are struggling to make these children recover nutritionally and thereby improve their overall outcome, including child development. But a lot more is, I think, still needed, and I think that's where you guys come in to try to help, address the knowledge gaps that are still there and try to take this to the next stage. And this was the end of my presentation. Thank you so much.
Thank you, Dr. Bansma. This was a very intriguing lecture. Thank you. Yeah, so I think we can now open the Q&A session, and you can just go ahead and unmute yourself and ask your questions. Maybe I can start with one while they get ready, Dr. Bansma. So these changes that we can induce with all of these different approaches that you described, is there any research that tell us how persistent these changes that we can induce, beneficial, hopefully, how they can persist in time? Or where are we with that?
Yeah, no, I think that's the golden question, right? There some early work, for example, in diabetics that showed that actually these changes, that was a fecal transplant, so basically whereby through an NG tube, diluted stool was given to people who were diabetic, and they looked at their microbiome changes over time, and they really regressed back after a couple of weeks back to their original microbiome. Because the microbiome is more dynamic, I think, than we initially appreciated, you would probably have to, keep introducing these interventions to change the microbiome permanently. But it's not true for everything, because for example, the antibiotic exposure seems to have much longer lasting effects. So there's a lot we don't know. Some changes seem to be shorter lasting, and other interventions seem to be longer lasting.
Yeah, thank you.
And maybe, yeah, go ahead.
No, go ahead. Go ahead. No, I was saying because
you know much more about this than I do.
No, this is not true, because these studies are in a younger population. Maybe, like, we really don't know. As you said, we need more research to see, and maybe their microbiomes would be more susceptible to change, or maybe not, especially if they are dysbiotic with these children with these enteric infections and altered environment, right? So.
Yeah, no, absolutely. Belinda, sorry.
Hi, I just had two questions. One is related to what Dr. Camilli just mentioned. So I was thinking, because like children are a lot more plasticable in terms of like it's the developmental period. So are there like specific ages where you have to test, perform these experiments, or like is there like, yeah, is there a standard for when you would start the intervention for it to last?
Yeah, so again, that's something we don't know. And Dr. Camilli and I are working on this together. Other perhaps periods in children's lives is during adolescence and puberty, where there's a lot of changes, hormonal changes, and changes in diet. And there's definitely changes in the microbiome that can occur during this period of time, and allows perhaps for modulation of the microbiome through interventions. So that might be another very critical window whereby there's more opportunity to affect the microbiome.
Okay,
but again, this is all still very early stages, so there's a lot we still don't know, especially in the intervention stage, right? A lot of the work is observational. A lot of the work is just single time points, so not really following up with multiple microbiome analysis over time. So I think in terms of design, there's a lot more that we can do to better understand, how these microbiome changes occur and how we can modulate them.
Okay, thank you. My other question was more out of curiosity for, I guess, research, because I know, like, at least for some other areas of research, it's usually very hard to get children to be approved to do experiments on children. So what are kind of like the challenges, or do you know if it's any different from, for example, if you had intervention on adults?
research in children is always more difficult. There's, I think, a growing recognition that, many interventions need to be replicated in children because children are not small adults and their bodies behave in very different ways. So the research community is arguing more and more for, even though it's challenging, but for ethical bodies to really appreciate the necessity of doing studies specifically in children to really improve the management of diseases, specific diseases in these populations. I don't think there's a big difference between doing research in children in high or low research settings. The ethical boards are very similar, and actually, in my case, there are multiple ethical boards, both in my case, in sick kids, and in these settings where the children are being recruited from. I think there are definitely differences. For example, in some lower resource settings, some caregivers are really very worried about blood sampling, which is less of a concern of parents in higher income settings. So a lot of it is also related, I think, to education and understanding of clinical research and what's being done with blood and how it's being used, making sure that privacy is protected, etc., and there's no harm done. So there are definitely some differences, especially I think related to the ability, the education level, for example, of caregivers. But we really do, I think, our utmost best to make sure that we are as ethical as possible.
Okay, thank you so much.
There's a question from Stephen.
Hi. Yes, I have a question. So I have a question about the micronutrients affecting the microbiome. So could you please go over how iron contributes to dysbiosis?
So iron is a nutrient of microbes. So the microbes use iron. So if you give iron supplementation, especially in large quantities, of course that micronutrient becomes very predominant in the gut, and then microbes starts to feed on them. They use them for their own metabolism and allows them to grow at a faster rate, and thereby causing dysbiosis. So there are two studies. One, I think, was done in one was done in Southeast Asia, and one was done in Eastern Africa. I think it was Zanzibar. I think it just recollecting. I think Zanzibar, showing, and the one was in a malarial population, the other was not, showing clear negative effects, including impact on mortality by giving iron supplementation, especially in children who are very vulnerable. So, for example, for the severely malnourished population, we don't give a lot of iron initially. So the diet that we use in that early critical phase is very low in iron, even though most of these children are very iron deficient, because we don't want to feed the microbes in the gut the iron and allow them to cause more enteropathy.
Thank you so much.
Thank you. There is a question in the chat, and then Laila. So the question in the chat is about, is asking if there are any mechanistic reasons that researchers thought that antibiotics would affect growth in the first place.
Yeah, again, it comes mostly from, I think, initially from the farming industry. Perhaps, Elena, you know this better than I do, how this all came about. What's the history of antibiotics and growth? Many decades ago, because I think it's a long-standing knowledge within that community, but I'm not sure exactly how it started. Do you know?
I think initially in the I also don't know much about the farm industry, but I think it started with wanting to control enteric infections, because when you grow animals like for feed, like for in the farm industry, and they get infected, especially pigs, for example, around weaning or soon after weaning, obviously this limits growth, and then they don't grow as well, and this is economically detrimental. And so they introduced antibiotics as a treatment against the infection. So not necessarily only enteric infection, but any infection that the animals could get in their early stages of life. So I think that's how it started. So it was like really like a therapy initially. So I think that's how it started. But yeah, definitely it's, but as Dr. Bansma said, right, this is what he has discussed is really where the research is right now. So unfortunately, we don't have many answers to your questions, because this is just where we are, and the studies that he described, I would say, like, are the key ones in the literature. There isn't that many more. So that's where we are with our knowledge. Yeah. Laila, did you have a question?
Yeah. So just following up on the question regarding the supplementation, has there been any negative associations with supplementing probiotics or prebiotics to children, similarly to iron or zinc?
No, not to children, but there is a study that was done in adults with pancreatitis, so inflammation of the pancreas, where they actually saw an increased mortality in the population that received through a nasogastric tube I think it was a probiotic. So that made people, clinicians, a bit more concerned, because it was always thought these probiotics are completely safe, but that really, I think, made people at least hesitate a little bit and avoid potentially in the highest risk population or in certain populations the use of probiotics.
Interesting. Okay, thank you.
So also I see many families in my different clinics where parents give probiotics to their children. It's quite commonly used, and it's probably okay for most kids, but I always tell my patients I cannot really completely rule out that there might be some negative impacts of probiotics, especially if a child's really vulnerable.
Thank you. We have two questions in the chat. One is about the relationship, let's say, between infection and growth. So if an individual has to focus on its immune response, for example, fighting an infection, then less energy is channeled towards skeletal growth. Is this something that is true, and what is the relationship there?
Yes, no, that's definitely true. Systemic inflammation leads to catabolism. So it affects that IGF-1, IGF-BP3 that access negatively. For one, it leads to, for example, insulin resistance to some degree, and that's through that pathway it impacts growth. Also, cytokines during inflammation impact appetite negatively, so you also start to consume less energy. So there are various pathways whereby inflammation impacts growth, either directly or indirectly by affecting appetite. Does that answer your question?
Shuyun, does it answer your question? Yes, it does. And then you also have a question about the plantarum. Sorry, I'm trying to think, what was that slide? Slide 44. Was this the slide with the Lactobacillus plantarum study in the germ-free mice? Yeah. So you're asking, what are the two strains of the plantarum? Are they the probiotic supplemented diet?
Yeah, so one was the natural strain and the other one was genetically altered, thereby affecting that particular microbe in such a way that it didn't have that growth effect anymore. I don't remember what the exact genetic change was, but it was genetically altered.
Yeah, thank you. Any other
And I think
questions? Oh, go ahead.
you have access to the PDF, right, of the presentation, so you can always look up the primary paper, that Cell paper that was where it was published in.
Yeah.
If you want to know more about it.
Yeah, for sure. Yes, they have the PDF. Any other questions? No? Okay, if not, we would like to thank you again, Dr. Bansma. I hope it is not too late over there. Thank you for your time, for answering all
African
these
bedtime.
questions.
9
Okay.
o'clock generally bedtime. So it's yes, it's time to go to bed. People wake up very early in Malawi. 5
Okay.
o'clock, 5.
Oh dear, yeah. Okay, we will not keep you any longer then. Thank you very much, Dr. Bansma.
Yeah, no, it was a pleasure. Thank you all, meeting you all, but I hope to do this again next year, but then in person.
Yes. Hope so too. Thank you. Thank you, Dr. Bansma.
Okay,
So everyone, we are getting like a 5 minutes break before we go into the second part of the lectures, which is about the enteric infections. Okay, so let's be back at around 2:25. Okay, everyone, so here we are. I hope everyone can hear me fine. Otherwise, let me know. I am going to start sharing. Can you see my slides okay?
Yes.
Yeah, okay, thank you, thank you. Okay, good. So for the second part of the lecture, we are going to discuss disruption of intestinal homeostasis and foodborne diseases. I'm going to quickly, yeah, okay, good. I just wanted to make sure that there was nothing in the chat. Okay. Good. So the first slides that I have today are for a quick recap of what we have learned so far. So I will go a little bit faster through them, but if you have questions, let me know. But I think you know most of these things already. But I just wanted to do like a quick summary so that we can put things into context before we discuss these enteric pathogens. Okay. So of course, you know the difference now between a eubiotic and a dysbiotic microbiota, where by eubiosis we intend good life, and therefore when the concept is applied to our intestinal ecosystem, this refers to the maintenance of an internal equilibrium in the spectrum of the bacteria, as opposed to a dysbiotic condition, which is characterized by qualitative and quantitative changes in the intestinal microbiota, their metabolic activity, and their distribution and localization. So, as you know, there are several mechanisms that underlie homeostasis in the intestinal tract. And this is a representation of an intestinal epithelial cell, which shows like the apical side, which is towards the lumen, and the basolateral side, the apical side covered by the mucus layer that we learned a lot about, and the microbiota residing on or within, embedded the outer layer of the mucus layer, and anyway towards the lumen. And then these mechanisms that are in place for the intestinal cells to maintain a situation of tolerance. For example, periodical sampling of pathogen-associated molecular motifs that are sampled by the epithelial cells and result in the expression of genes in the epithelial cells that maintain a significant amount of T regulatory cells in the lamina propria, and therefore prevent the activation of the inflammatory pathways, right? This is all what you have learned so far. But so this intestinal homeostasis depends on several mechanisms, the secretion of the antimicrobial peptides in the lumen that help to keep it under control, the number of commensals, but also potential pathogens, the epithelial barrier, the rapid repair of epithelial defects, the downregulation of the innate and adaptive immune responses, and the killing of bacteria that may translocate across the intestinal monolayer. And for those who are interested, just because we never really mentioned restitution and regeneration, I have added this slide that explains a little bit with what these are. So when there is a damage in the intestinal epithelium, restitution refers to the initial repair to this damage. It occurs within minutes to hours and does not really require cellular proliferation. So there are local epithelial cells that migrate towards the wound to restore the integrity and the physical barrier. On the other hand, the regeneration refers to the proliferation of epithelial cells, which is stimulated and which starts happening in the crypts that are localized near to the wound, and in order to replenish the cell pool and also reestablish the integrity. So just a little parenthesis because we have never mentioned this. Okay, so this is what the host has available in order to maintain homeostasis and this situation of equilibrium when there is no inflammatory response to the bacteria in the lumen, either commensal or exogenous that may be ingested. So which are the 4 potential mechanisms, on the other hand, by which dysenteric bacteria and fungi can induce chronic immune-mediated intestinal injury? Okay, so there are 4 essential routes. So there could be like a pathogenic infection, which is what we will discuss today. There could be a dysbiotic microbiota for many, many different reasons, some of which you have heard earlier from Dr. Baansma. There could be a defective containment of commensals and defective host immune regulation. So in terms of the pathogen bacteria, the exposure to a pathogen or to a functionally altered commensal microorganism can induce an increased stimulation of the immune response. It may be the pathogen itself, it may be a toxin that they gain access to the epithelium and then they generate an inflammatory response. But the homeostasis can also be challenged by dysbiosis. So an abnormal composition of the gut microbiota, which may be associated with increased permeability, because this dysbiosis typically is accompanied by a reduction in the number of microorganisms that can produce butyrate through their fermentation processes. And as we know, butyrate is beneficial for the health of the colonocytes, is their main energy source. It's important for maintaining the barrier function. And usually this is also accompanied by an increase in the numbers of bacteria that produce aggressive molecules such as, for example, H2S, that would be detrimental to the intestinal ecosystem. On the host side, there could also be defects in the ability of the host to contain the commensal bacteria. So maybe a situation where there is no pathogen necessarily, no pathogens that are ingested, or not necessarily an altered composition in the microbiota, but an altered capacity of the host to handle such a large community of microorganisms. So for example, this could be the result of defective secretion of antimicrobial peptides or IgA that would result in bacterial overgrowth. And if there is bacterial overgrowth, there may also be a defective killing of pathogens, and both can lead to an increase in the bacterial antigen load and therefore a lack or a diminished capacity of the host to handle this complex microbiota. And finally, there can be defects in the host capacity to regulate their immune response. There could be a defective downregulation of innate immune response, for example, that can induce an inflammatory response and the secretion of pro-inflammatory cytokines that then would contribute to the perpetration of inflammation. So as I said, so this was like our general recap and summary of what we know so far. And as you saw, that pathogens and the appearance or the fact that pathogens can reach the intestinal community down in the small intestine and in the colon is one of the ways through which homeostasis can be altered and inflammation can ensue. So today, specifically, we will discuss pathogens that are ingested with food or water, so foodborne illnesses. These pathogens are also called, also known to call food poisoning as an alternate terminology. And these illnesses, these diseases, mainly result from our failure to control an hazard, which can be an identified hazard or a non-identified hazard, but that would be the consequence. So a hazard, if we think of it in its simplest form, it's something that has the potential to cause harm, right? So, for example, in food and water, a hazard would be something that make food and water unfit for human consumption. So there may be like many different causes, the major being biological, chemical, and physical causes. So for the biological causes, which are the topic of this part of the lecture today, this may include pathogenic viruses, bacteria, protozoa, parasites, and fungi, which contaminate the food or water that we may eat or drink. But there could be illnesses that are foodborne and that are caused by different types of hazard, like chemicals, pesticides, cleaners, residues that are found in the food that result from the production process, as well as a physical hazard like foreign objects like glass, bone fragments, pebbles that can find their way to the finally packaged food. So what are the common symptoms of these biological foodborne or waterborne illnesses, which are the ones that we want to focus on today? So these can have, usually these biological illnesses result in nausea, vomiting, fever, diarrhea, abdominal pain, chills. Sometimes they are confused with flu, or they are called abdominal flu. So these are the general, but obviously not all pathogens produce the same effects, but generally speaking, these are the common symptoms. So these common symptoms that we just mentioned are the ones that appear in the immediate, right? As soon as these pathogens become infects, and then the symptoms manifest relatively soon. But unfortunately, these diseases also have, or may have, not in every individual, but often they can have complications and effects that are long-term. So, of course, they could result in dehydration, especially if they are accompanied by diarrhea and this type of symptoms. But more worrying long-term effects may also ensue. And the ones that are the most known and most common include hemolytic uremic syndrome, or HUS for short, which is a consequence of infection with E. coli toxins, or enteromorrhagic E. coli, which may destroy the red blood cells and the lining of the blood vessels. We'll see later in a slide a little bit more in depth. As a consequence of a Campylobacter jejuni infection or Salmonella, there could be long-term inflammation of the joints and reactive arthritis. There could also be irritable bowel syndrome as a long-term consequence of infection with different kinds of enteric pathogens. And following Campylobacter jejuni infection, there could be the manifestation of the Guillain-Barré syndrome, which causes muscle weakness and may even result in paralysis. So when we think of these pathogens, although most commonly, luckily, they have like acute effects and symptoms that would resolve within a few days, unfortunately, in many cases, they have long-term effects, and which also contribute to the burden of these infections. So in the next few slides, I'm going to give you like a few facts about this infection, especially in Canada, and what systems are available in Canada to keep these infections under control. So obviously, I'm not expecting you to learn all of the data that you will see in those tables and in those slides. It's more for your general knowledge to know how this is managed in our country. So Health Canada, first of all, like the importance of the topic, right? So according to Health Canada, there's an estimate that every year, 1 in 8 Canadians, meaning 4 million people, suffer from illnesses caused by foodborne bacteria. Right? So if you come across the publication on the epidemiology of these diseases, or if you refer to the websites from Health Canada, the Canadian Institute for Health Information, the Canadian Food Inspection Agency, you may see these data expressed as prevalence or as incidence. So prevalence would measure how much of a disease there is in a population at a given point in time, right? At a specific point in time. So we calculate the prevalence by dividing the number of persons with the disease at this time that we are looking at by the number of individuals that are examined. And usually it's a rate that is expressed per 100,000 people. The incidence, on the other hand, measures the rate of occurrence of new cases in a given time period. Okay. So again, I mentioned that there are several agencies that work in concert, and they're kind of complementary in Canada to keep these diseases documented, and also run analysis to look at trends and longitudinal analysis throughout the years in order to learn how these diseases can be kept under control, what are the major sources, how intervention can be done, and what could be done to prevent them, especially from happening. Right. So, for example, in Canada, we have the Center for Foodborne, Environmental, and Zoonotic Infectious Diseases, the National Enteric Surveillance Program, and the National Notifiable Diseases Databases. So, for those who are interested, I put the website here. So they work together. Some data that they collect overlap, but not necessarily, right? So all those numbers that you see at the Health Canada website, or if you're interested, which is the pathogen causing the highest number of infection in a given year, they are produced based on the data that are collected by these, by all of these agencies and programs. Okay, so how do we define an enteric outbreak? You probably have heard sometimes in the news, unfortunately, this happens, and there may be like a food recall. So what is an enteric outbreak? This is the occurrence of 2 or more cases of an enteric illness that are linked in terms of time, exposure to the source, and most often location. So we'll see a few examples of enteric outbreaks. So this is an example of how data could be presented if you go to the Health Canada website. So for example, this referred to 2003. It gives you like the breakdown by the different type of agents. So here we have viruses, bacterial agents, parasites, and it gives the number of outbreaks defined as we saw earlier, and the number of cases, individuals affected. So for example, in 2003, most of the outbreaks were due to viruses, with norovirus being like the predominant cause, while among bacterial illnesses, Campylobacter was the main cause. So unfortunately, many of these enteric infection and diseases go unrecognized or are unreported, because on one side, because luckily maybe they don't cause very serious effects, and therefore an infected person may not even go and see their doctor, and therefore this remains unreported. Sometimes it is deemed not necessary to follow up with an investigation to determine which is the pathogen that has infected the person, because maybe it's not cost effective and the person will be treated. It's not necessary to determine treatment. So there would be an infection which is unrecognized, like we wouldn't know which pathogen caused it. And so the estimate that we have is that at least, yes, is that only 1 to 10% of the cases are in fact reported. And as one may expect, obviously, across the population, individuals who are more at risk are the elderly, children, and immune-compromised individuals. So I said about the incidence and the importance of how of these diseases in Canada, and how when we examine the burden of this disease, we also have to consider the long-term consequences. So it has been estimated that in 2 to 3% of the cases, these foodborne illnesses can result in chronic health problems that include the hemolytic uremic syndrome that I mentioned earlier, which ultimately could lead to kidney failure, and other conditions such as chronic arthritis. So Health Canada estimates that the annual cost related to these illnesses and deaths is between 12 and $14 billion. So essentially, there are two types of foodborne illnesses. There is the food infections and there is the food intoxication. So the difference lays in the fact that food infections are caused by the ingestion of living pathogens. So the food or the water that are ingested contain the living pathogen in them. So for example, Campylobacter jejuni, Listeria monocytogenes, E. coli, Salmonellosis. These are examples of food infections. Food intoxication, on the other hand, refers to the ingestion of food which contains the toxin that is produced by the pathogen. So the pathogen may not necessarily be there anymore as a live pathogen, but the toxin is present and would cause the illness. So many different causes of foodborne illness across the entire spectrum of the microorganisms, including bacteria, molds, viruses, worm, protozoa, and also prions. But in Canada, as this infographic shows, there are 5 major culprits. And these are the ones shown here. So the norovirus, which is the leading cause of foodborne illnesses and hospitalization in Canada. Listeria, which is the leading cause of deaths related to foodborne illness every year. Salmonella, which contributes to 1 to 4 hospitalization among all of the foodborne illnesses. E. coli 0157, which is one of the top foodborne bacteria causing severe illness, which can be accompanied by the long-term hemolytic uremic syndrome, which was mentioned earlier, and Campylobacter, which is the 3rd leading cause of foodborne illness and hospitalizations. So now we're going a little bit more in detail into how these 5 pathogens can cause disease. So let's start with Campylobacter jejuni, and the disease that it causes is called campylobacteriosis. So Campylobacter is a microaerophile microorganism. So it requires oxygen to survive, but lower levels of oxygen that are present in the atmosphere. Okay, so this is the leading cause of bacterial foodborne diarrheal disease when we consider the entire world. Okay, and it is typically found in foods like raw or undercooked poultry, essentially beef and pork, non-chlorinated water, raw milk, which has not been heat treated or filtered, infected animal or human feces, cattle, puppies, kittens, pet birds, and the reservoir is food and environment in developing countries and food production animals in the developed countries. So poultry products are essentially the main cause of this foodborne illness worldwide. So Campylobacter jejuni has a very low infectious dose. Just 500 to 800 cells are sufficient to induce the disease. And actually, this was proven experimentally by a doctor in the UK in 1981. He voluntarily ingested 500 cells of Campylobacter jejuni to prove that it is the cause of campylobacteriosis and that such a low infectious dose was sufficient to cause the disease. So the signs of campylobacteriosis are diarrhea, with or without blood in the stools, cramps, abdominal cramps, fever, nausea, and it may affect all age groups, but in developing countries, it is largely a pediatric disease. So just to have an idea of the trends of campylobacteriosis in Canada and Ontario, these were the numbers in that time period between 2005 and 2014. Obviously, you don't have to learn these numbers, okay? And so Campylobacter jejuni, unfortunately, can come with a long-term consequence, which is a condition known as Guillain-Barré syndrome. So this is an autoimmune disease. So the host produces antibodies against Campylobacter, and specifically against gangliosides that are produced by the bacterium. These gangliosides are very similar to, or are a mimic of, the gangliosides that the host expresses in their own tissues, including peripheral nerve tissue. So this results in this form of autoimmune disease, which can cause nerve damage and lead to muscle weakness and eventually paralysis and even death. So the Guillain-Barré syndrome usually develops 1 to 3 weeks after the Campylobacter jejuni infection. The other pathogens that I wanted to discuss is Salmonella species, which causes salmonellosis. So the Salmonella species and Serovars, these are all gram-negative bacteria, non-spore forming. They are rods, as you can see in the picture, and they belong to the family of the Enterobacteriaceae. So essentially, salmonellosis can manifest as 2 different types of disease depending on the infectious agent. So one type is the acute gastroenteritis, which is caused by essentially Salmonella enteritidis. And the other one is the enteric fever, which is also known as typhoid, and which is caused by Salmonella typhi. The difference between the two is that enteric fever is a systemic disease because the Salmonella, the bacteria, invade the bloodstream. So obviously, as you can imagine, the outcomes is much worse with the enteric fever. So the Salmonella is essentially the favorite habitat of the Salmonella is the intestinal tract. It can be found in the environment, but it wouldn't be able to reproduce that quickly. So the intestinal tract is really the main reservoir and preferred habitat. So hence the contamination of food and water with Salmonella is essentially the fecal oral route. So again, it is common in poultry products and eggs, raw milk, raw beef, unwashed fruits and vegetables, or that are washed with contaminated water. And it has been found that also reptile pets like snakes, turtles, and lizards can be sources of contamination. So the signs are diarrhea, fever, and cramps, and the gastroenteritis form, so the acute gastroenteritis, has a quick onset, about 12 to 72 hours, and lasts for a week. So in Canada, in 1998, there was an outbreak of Salmonella enteritidis, which still remains one of the largest foodborne disease outbreaks in Canadian history. There were more than 800 reported cases, and the cause, I think, was down to packed lunches. So it was really one of the largest outbreaks in the history of Canada. So as you can see from the table here, of all the serotypes of Salmonella that can cause salmonellosis, the most common is by far the enteritidis, which is the one that causes acute gastroenteritis. So this is an example of what you would see as a communication from Health Canada and the Canadian Food Inspection Agency when there is an outbreak of an enteric pathogen or a foodborne pathogen, right? In this case, it referred to an outbreak of Salmonella infections linked to poultry, including frozen raw, breaded chicken products. It was in 2018. You don't have to learn all of this text, but usually an investigation is run, and at the end, there is a summary of the investigation with what has been found, if the source has been identified, and which procedures have been put in place, which may include recalling the food that was found to be contaminated, of course, and the number of cases and where these cases were identified. Right? So this is an example of a report that the Canadian Food Inspection Agency would produce at the end of an outbreak investigation. And all of this is publicly available material that you can find accessing their website. Okay, so Salmonella enteritidis is the serovar that causes acute gastroenteritis. So just to reiterate a bit, the most common food sources are eggs and poultry. Raw milk, pork, beef sprouts, and raw almonds are less common. International travel, if for contact with reptiles, can also play a role. And this disease manifests with fever, abdominal cramps, and diarrhea that begin relatively soon following the infection. And usually the infected individuals would recover without an antibiotic treatment. But if the diarrhea is severe, there is risk for dehydration, then the individual must have to be hospitalized. On the other hand, Salmonella typhi is the cause of the systemic form of infection. Now, humans are the only reservoir for this bacterium. So it is carried in the bloodstream and in the intestinal tract. So it would gain access to the bloodstream by breaching through the intestinal barrier. And then it is shed in the feces. So this is how food is contaminated, through handling, non-proper handling, or through sewage, or being processed with contaminated water. So although this infection can be treated with antibiotics, typhoid fever, which ensues, is life-threatening. So the symptoms here are fever, stomach pain, weakness, headache, loss of appetite, and diarrhea is not very common in this case. So, and this table just summarizes what we have said so far about the differences in these two diseases. So how does Salmonella gain access to the circulation? Because as for any other pathogen, right, the host has put in place a various system to block the pathogens and prevent infections, right? Starting with the low pH in the stomach and the production of the digestive enzymes and the bile acids, which should keep these bacteria under control. But of course, these bacteria also have developed strategies that allow them to escape these innate mechanisms of response. So how does Salmonella gains access to the circulation, or how does it infect intestinal cells? So there are essentially three mechanisms that have been proposed. So the first one is invasion of the enterocytes. The second one is through invasion of the M cells that are found interspersed in between the enterocytes along the epithelium lining. And the third is through dendritic cells that can protrude in between adjacent enterocytes and sample bacteria in the lumen. So in this case, they would sample Salmonella cells. So upon infection, the Salmonella it translocates across the monolayer, and they would colonize the Peyer's patches and the mesenteric lymph nodes, and then lead to the development of the immune reaction. So in the case of the non-typhoidal serovars, like the enteritidis, there is a local inflammatory response that ensues. Okay, there can be leukocytes infiltration and diarrhea, but the disease remains localized to the intestinal tract. In the case of the enteric fever serovars, so like the Salmonella typhi, the bacteria are able to enter the macrophages and then to disseminate the illness systemically. The other pathogens, which is one of the main 5 culprits in Canada, is E. coli or 157H7. So E. coli, as we know now, they are gram-negatives, they are not spore-forming, they're also rods, and they also belong to the family of the Enterobacteriaceae. Right? So generally speaking, E. coli is a common inhabitant of the intestinal tract. Okay, it is a commensal, and it's also useful for the intestinal tract homeostasis, because, for example, it suppresses the growth of harmful bacterial species and it produces vitamins. But there are a few strains of E. coli, like the O157H7, which are pathogenic. So these numbers and letters refer to their antigen, to the type of antigen that they carry. Okay, so the O refers to the lipopolysaccharide component, and the H refers to the flagellar antigen. So as I mentioned earlier, 157H7 is a primary cause of hemorrhagic colitis, and long-term can result in hemolytic uremic syndrome and acute kidney failure in children. And this is through the production of a toxin, which is called verotoxin or Shiga toxin. It is also called Shiga toxin because this is the exact same toxin that another pathogen called Shigella produces. Okay, so both the Shigella and the E. coli 157H7 produce the same toxin, which may result in hemolytic uremic syndrome. So which are the causes, the sources of E. coli infection? So essentially it's undercooked meat, salami, alfalfa sprouts, not properly washed lettuce, so fresh vegetables, non-pasteurized milk, contaminated well water. Signs: watery or bloody diarrhea, nausea and cramps, and again, relatively quick onset within 2 to 5 days from infection. So I mentioned the Shiga toxin or verotoxin that E. coli O157H7 and Shigella dysentery produce, and which is at the basis of its pathogenicity mechanisms. So a few details about this toxin. It is a protein of 68 kilodalton that consists of 2 subunits, a subunit A and a subunit B. So the B subunit is the one that first initially binds to the globocyte 3 receptor expressed on the host cells, but particularly the small blood vessels, so not the large arteries, right? So the small blood vessels, which are found in the intestine, they are found in the lungs, and that they are found in the glomerulus in the kidney. So by binding to this receptor, the B subunits mediates the entrance in the cell. Once it's inside the cell, the A subunit inactivates the ribosomes and stops protein synthesis. So the consequence is the death of the cell. So because these are cells of the vessels, so the vascular epithelium, the breakdown of the vessel lining is what results. And in the intestine, this leads to hemorrhage, so blood in the stools. On the other hand, in the kidney, when this happens in the glomerulus, which is the filtrating unit in the kidney, this leads to kidney failure. So these are some epidemiology about the incidence of verotoxin producing E. coli in Ontario and in Canada. So for example, in 2014, there were 127 confirmed cases in Ontario. So one may think that this is a low incidence, but when you look at the consequences and also that it may result in death of the infected person, then this is when you realize that how important it is to keep this pathogenic infection under control. And a relatively recent outbreak was in Canada and the US in 2017 and 18. This was linked to a lettuce product. And again, similarly to what I showed earlier, this was the update on the final investigation, like the summary on the final investigation that was conducted by Health Canada about this outbreak. So you may be familiar with the Walkerton tragedy, which happened in 2000 in the city of Walkerton, which, as it is shown here in the map, it's about 180 kilometers northwest of Toronto. So in that month of the year 2000, there was like heavy rainfall that washed away the livestock fecal residue, which ultimately contaminated the municipal water supply. So unfortunately, the water supply was not properly chlorinated, which resulted in the municipal water being contaminated with E. coli 0157H7, so the one that we just discussed, but also Campylobacter jejuni and other pathogens. So the residents were drinking and using contaminated water. So 2,300 people had developed bacterial gastroenteritis. 27 of them developed the hemolytic uremic syndrome, and there were 7 deaths. So not only this burden, but after 2 years, research continued, because now this city and this event became the subject of of extensive research in order to understand how could this have happened, how can we prevent this from happening again in the future, and also how can we help those people. And it also allowed us to learn a lot about enteric infection and the long-term consequences. And in fact, after 2 years, in this population, it was found that there was a 3-fold higher risk of developing post-infectious irritable bowel syndrome. And so highlighting a significant relationship between acute enteric infection and subsequent irritable bowel syndrome. Now, Listeria monocytogenes, which causes a disease which is called listeriosis. The listeria is a gram-positive rod, and its habitat, in fact, is decomposing organic matter. So this bacterium grows at low temperatures, so it would survive in the refrigerator. So how can we get infected with this bacterium? So again, it's unpasteurized milk and dairy products that are obtained from unpasteurized milk, such as many different cheeses, vegetables that are not properly washed, and ready-to-eat meats are a main source of Listeria monocytogenes infection. So like deli meat, spreads, hot dogs, and so on. So the signs and the way this disease develops are a bit different. So the signs are still, we have flu-like symptoms like nausea, vomiting, cramps, and diarrhea, but unfortunately listeriosis, especially in pregnant women, can lead to abortion and stillbirths. And it can also lead to a systemic disease like septicemia, which can lead to death. So there was an outbreak in Canada in 2008. These are the numbers. There were 41 confirmed cases in Ontario, but there were confirmed cases across many of the other provinces and 23 deaths in total. So, and this, I think, was traced back to a deli meat product. So how does it work in terms of the infection routes and pathophysiology of the Listeria infection? So again, the bacterium has to gain access to the intestinal tract. So it has to survive passage through the stomach and the more proximal region of the small intestine. It is still controversial if it invades through the M cells or through the enterocytes, but there is intestinal translocation happens, and the bacterium gains access to the circulation. After this, it is thought that it is found as a first step in the liver, and that it multiplies within the hepatocytes before spreading throughout the circulation and causing septicemia. So now in adults, listeriosis can result in meningoencephalitis. So it can affect, it can lead to infection in the brain, in the central nervous system, and resulting ultimately in paralysis. But as I mentioned earlier, it can get in pregnant women, it may gain access through the fetus and result in abortion or stillbirth. And finally, the last agent that I wanted to mention is the noroviruses. So it is plural because there are many different viruses that all belong to the norovirus and have different, a few variation depending on the geographical region where the outbreak is coming from, but they're usually all studied together and called Norwalk-like viruses from the city of Norwalk, where there was an important spread and outbreak of noroviruses in 1968. So these viruses are the most important cause of non-bacterial epidemics of acute gastroenteritis across all ages. They are very common though, for example, in child cares or also in nursing homes and cruise ships. The humans are the essential reservoir for these noroviruses, and therefore the fecal-oral route of spreading is the route of infection, of transmission, person to person. So they have a very small infection dose. About 100 viral particles are sufficient to for the infection. And the signs are the usual that we mentioned so far. So nausea, vomiting, diarrhea, and abdominal cramps. Usually it has a very short duration, and within 2 days it resolves. So I mentioned that in Canada, Health Canada and the Canadian Food Inspection Agency overlook and make sure that the food that we eat is safe. We have in Canada, this is one of the safest systems in the world, but still it is not perfect, and sometimes this infection may occur. So if you go to the Canadian Food Inspection Agency website, which I put the link here at the bottom of the slide, you can look in real time what's going on currently right now in Canada. So this is a picture from the homepage of that website last year around this time. So obviously there was the coronavirus outbreak update, and then there was a list of the current food recalls that were going on at the time. Yesterday, I went and checked what do we have now on that webpage. So there is the latest is about avian influenza detections, and then if you scroll down, you have the specific details about food recalls, food infections, food safety, importing food, plants, and animals, and what is happening currently in terms of outbreaks or any foods being recalled right now from the Canadian market. So I just wanted to give you like a few information about what a food safety investigation recall process is in Canada. So it essentially goes along 5 steps. The first step refers to the triggers. So what starts a food safety investigation, right? There could be an inspection, there could be a consumer complaint, it could be an industry complaint. It is a foodborne illness outbreak. So this starts the investigation from CFIA. The food safety investigation, which is step 2, involves many different steps, and it's complex. It has to determine the cause, the risk, assess the magnitude of the risk, determine if it's necessary to proceed with the food recall, and so on and so forth. So it refers to investigating the sources of where the contamination may be coming from, where it could distribute, like who, to whom is available this product, like which categories, for how long, and it may refer to having to do an inspection on site, for example, in a production plant or in grocery stores, to see if the food has been properly stored and so on. The third step typically is a health risk assessment. So this, following the investigation, we need to determine if this risk is a potential harm for the population. And what do we need to assess and do in order to decide how to mitigate this risk, like which action we need to put in place. The step 4 is the recall process, when it is determined that the food needs to be removed from the grocery stores and people cannot consume it. So there is a food recall, which is the removal of the food from further sale and use. So obviously this implies implementing the recall, informing the public, and then verify that the recall has happened. And the last step is the follow-up. Once the recall is complete and there is no more of these food products in the marketplace, the CFIA will continue to work with the manufacturer, the importers, to see which problems may have led to this and how they can be resolved to avoid a similar problem in future. So in terms of the recall process itself, like there are 3 main categories. So the category 3 is the less worrisome. It represents a situation in where there is a reasonable probability that the consumption of that food is not likely to result in an adverse health consequence. And then class 2 and class 1 food recalls are more concerning. So with class 2, there is a temporary or not life-threatening health consequence that may happen upon consumption or exposure to that food product. And in class 1, this represents a situation where there is a probability that the food, consumption of this food will lead to adverse health consequences, which are serious, could even be life-threatening, and the probability of the foodborne outbreak situation is considered high. So this is to say that there is a system in place in Canada, and this is how it works, and it's very open and transparent. So by going to the website that I put the link, you can have all of the information on what's currently going on and the results of the investigation, what they led to. And again, these are a few websites for those of you who are interested in going a little bit more in depth into this. So I am going to stop sharing and I'm going to ask if there are any questions. No questions? Okay, so if there are no questions, then this ends our lecture today. If more questions come up, put them please in the discussion board. Okay, so thank you, and I will see you next week in person. Bye everyone.